Good afternoon, biology students. We are currently on Chapter 7, Section 3, and we're going to discuss structures and organelles. And as we're discussing it or you're watching videos, I want you to think of a city, okay? Who carries back and forth information from one person to the next? The mailman, okay? And what organelle inside of a cell carries and transports information throughout the cell? It's called a Golgi apparatus or a Golgi body, okay? So these are things that you need to be thinking of because you're going to have quite a large project based on this, and you will be presenting in front of the class. So, main idea, eukaryotic cells contain organelles that allow the specializations and the separation of functions within the cell. So cytoplasm and cytoskeleton. The environment enclosed by the plasma membrane is a semi-fluid material called cytoplasm. In prokaryotes, all the chemical processes of the cell take place directly in the cytoplasm. But in eukaryotes, these processes take place within organelles inside the cytoplasm. Cytoskeleton is a supporting network of long, thin protein fibers that form a framework for the cell and provide an anchor for the organelles. I think that should have been provide an anchor for the organelles. So if you think of a skeleton for a body, it provides the framework, okay? So microtubules are long, hollow protein cylinders that form a rigid skeleton for the cell and assist in moving substances within the cell. Microfilaments are the thin protein threads that help give the cell shape and enable the part or the entire cell to move.
Well, you know our style. We love our science with a side of comics. So we want to take you on a tour of the ride of your life, into the inside of a cell. Now, to start our trip, we're going to have to get through this cell membrane, also called a plasma membrane. It's selectively permeable, which means it'll only let certain select materials in and out. But by doing so, it keeps things in the cell stable, also known as keeping homeostasis. And we have an entire video on just the membrane itself, which is found in all cells. But for now, we're just going to have to squeeze through this protein in the membrane. Now inside the cell, we find ourselves in this jelly-like material called cytoplasm. It surrounds all these internal cell structures, and you'll find it inside both prokaryotes and eukaryotes. Now, organelles that are floating around in the cytoplasm can have more support than you might think. Cells contain a cytoskeleton, which is a collection of fibers that can provide support for the cell and its organelles. And the cytoskeleton can even play a major role in cell movement. The cytoskeleton actually deserves its own video, because it's very complex, and its organization can vary depending on what kind of cell you're looking at.
So the nucleus. Next to the nucleus, you need to write the textbook page number that is located for the nucleus. Contains most of the cell's DNA, not all, which stores information used to make proteins that determine a cell's growth, function, and reproduction. Basically, it's the set of directions to the next human being, okay? It's surrounded by a double membrane called a nuclear envelope. Ribosomes are organelles that manufacture proteins. That's what they make. They're like little factories, okay? So they're not membrane bound like other organelles, and they are produced inside the nucleus in the nucleolus. Endoplasmic reticulum, otherwise known as ER, is a membrane system of folded sacs and interconnected channels that serves as the site for protein and lipid synthesis. There are two types, rough and smooth.
Through the cytoplasm, let's start with ribosomes. They are not membrane-bound organelles, and they're going to be in both prokaryotes and eukaryotes. They make protein, which is really important because that's what so much of genetic material, DNA, codes for, protein.
We're going to come back to this for the DNA portion of our unit. When I say this is not going away, I totally mean it. And you're going to have to be building this also by yourself. And as a class, we're going to have many, many labs. And it's 30% of your overall grade. So why not just learn it now?
Ribosomes can be free in the cytoplasm. But they can be attached to another organelle too, which we'll talk about a little bit later. We're now going to focus on organelles that will be membrane bound. So we're going to be focusing on organelles that you would find in a eukaryote cell. This takes our travel to the big boss, the nucleus. Now in eukaryotes, it holds the genetic material. Genetic material as in DNA, for example. Both prokaryotes and eukaryotes have DNA, but if you're a eukaryote, you have a nucleus to put it in. And the nucleus controls the cell's activities, and inside it, it has a nucleolus, which is where ribosomes can be produced. Attached to the membrane of the nucleus, or nuclear membrane, you will find the endoplasmic reticulum, ER for short. It does a lot of processing of molecules for the cell, like protein folding, and it's highly involved in actually transporting those molecules around, like a highway. And there's a rough ER, which has ribosomes attached to it, making it, as you can imagine, rough, and then smooth ER, which doesn't have the ribosomes. Rough ER specifically tends to be involved with protein producing and transporting, because remember that ribosomes make protein. Now, molecules that leave the ER can be sent away in vesicles that actually pinch off the ER themselves. Now, smooth ER has many additional roles, including detoxification, which is one reason why your liver cells tend to have a lot of smooth ER. And another additional role of smooth ER is that it can make some types of lipids.
The Golgi apparatus is a flattened stack of membranes that modifies, sorts, and packages proteins. Proteins are packaged into sacs called vesicles, which can fuse to the plasma membrane and release the proteins to the environment. A vacuole is a membrane-bound sac used for temporary storage. It can store food, enzymes, or even waste. Vacuoles are also found in plant cells in addition to animal cells.
Next, the Golgi apparatus. It's the ultimate packaging center. It can receive items from the transport vesicles that pinched off the ER. It has enzymes that can modify molecules it may receive and sorts the material it receives as well. It can determine where to send those molecules, including some that may eventually be sent to the membrane, so they can be secreted, which means items that can be sent out of the cell. So with all that's going on in here, you might start to wonder, what's powering this thing?
Moving on to lysosomes. These are vesicles that contain substances that digest excessive or worn out organelles and food particles. Think of it as the cell or organelle graveyard. They also digest bacteria or viruses that can invade a cell. Centrioles are organelles made of microtubules that function during cell division. They're located in the cytoplasm. Mitochondria convert fuel particles into usable energy. They have an outer membrane and a highly folded inner membrane that provides surface area for breaking the bonds in sugar molecules. And they're right here. You could probably see it in your book better, but it kind of reminds me of a kidney bean.
So with all that's going on in here, you might start to wonder, what's powering this thing? The mighty mitochondria, or mitochondrion, if you're just talking about one. They're like power plants. The mitochondria make ATP energy in a process called cellular respiration. Now, it's not the type of power plant that you might think of. It runs on glucose, which is a sugar, and it needs the presence of oxygen to efficiently make ATP energy. Now, at this point, we need to mention that eukaryotes are not a one-size-fits-all. Animal cells can have differences from plant cells. We have a fork in the road here. For example, plant cells not only have mitochondria, but they can also have these awesome organelles called chloroplasts. Chloroplasts actually make glucose by using light energy in a process known as photosynthesis. They tend to even have a green look to them because they have a pigment that captures the light energy and it reflects green light. Now, both plant and animal cells can have vacuoles. Now, vacuoles can have a lot of different functions, but many types act as storage of materials. Plant cells can have one large vacuole called a central vacuole, while animal cells can have smaller, several vacuoles. Now, remember how we already said that all cells have membranes? They do. But plant cells additionally can have a cell wall, which is a layer that offers additional protection and shape maintenance that animal cells do not. Okay, now how do we get out of this animal cell that we've been in?
Plant cells and some other eukaryotes contain chloroplasts, which capture light and convert it into chemical energy in a process called photosynthesis. They are composed of multiple small discs called thylakoids, which contain the light-capturing pigment chlorophyll.
Well, we could get out like a protein would. So if we were a protein, we would only be made because of instructions from DNA. And remember that in eukaryotes, DNA is found in the nucleus. We would be made by a ribosome because ribosomes make protein. The ribosomes could be attached to the rough ER, and the rough ER highway would provide a vesicle to send us to the Golgi apparatus where sorting can take place. And if we're tagged for being secreted, we could be sent off through a vesicle from the Golgi to the membrane. Out we go. Now just keep in mind that in our quick tour, there are still so many more awesome organelles found in lots of different types of eukaryote cells to continue exploring, so to the Google for more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Now, something a plant cell has that an animal cell does not have is a cell wall. It goes on the outside of the cell membrane, and it's a thick, rigid mesh of fibers that surround the outside of the plasma membrane. It protects the cell and gives it a structure, and it's made from carbohydrates called cellulose. Now, a couple of things the video didn't go over is cilia and flagella. Cilia are short, numerous projections that look like hair. They move in tandem like oars in a rowboat. Row, row, kind of how it happens. Flagella, on the other hand, are longer and less numerous than cilia, and they move with a whip-like motion. Both are made of microtubules. So features that plant cells have that animal cells generally don't are chloroplast and chlorophyll, vacuoles, and cell walls. So organelles at work. After writing this, put the page of your book. An understanding of different organelles allows for an understanding of cellular processes. The first one, super important here because we're going to be visiting this for like the next 5 months, not kidding. Protein synthesis. It begins in the nucleus with the information contained in DNA. RNA and ribosomes leave the nucleus and produce a protein on the endoplasmic reticulum. Proteins produced in the endoplasmic reticulum are sent to Golgi apparatuses for packaging. The packaged proteins are then delivered to other organelles where they serve a variety of functions. At this point in the game, I would highly suggest that you start making some flashcards now and practice. I would say at least half of your EOC at the end of the school year will be about everything we've been going over since Chapter 6 through the rest of the school year. Okay, so that's my first suggestion to you. Second one, don't forget, we will be having some pop quizzes. Great way to earn some bonus points. There's no need to draw out the cell. I'm actually going to be giving you 2 different cells, an animal and a plant cell, and you will be labeling those and coloring those. It's part of our in-class work that we'll be doing. So just let me give you the copied version. It's a little bit nicer for those of us who can only draw stick people.